and welcome to this lesson for higher biology, which today is going to be looking at metabolic rate. So prior to now, we have been looking at respiration and the fact that chemical aerobic respiration is the process by which cells are producing ATP energy. That ATP energy is used in those cells in order to carry out the reactions of the metabolic pathways, and consequently provides us with what we refer to as the metabolic rate of an organism, how fast all of those reactions are actually happening. And that's what we're going to focus on just now, before we move on to the next section, which will look at conformers and regulators, which are essentially the different types of organisms and how they are managing to maintain their metabolic rate depending on their particular physical differences. So primarily today what we're looking at is how metabolic rate can be measured, and we're also going to look at the delivery of oxygen to cells within different organisms, which helps maintain that metabolic rate, providing the oxygen for aerobic respiration. So what you need to be able to do is list the possible measurements used to compare metabolic rates. You need to be able to explain why different organisms have different metabolic rates. You need to be able to name and describe the different circulatory systems used by different organisms and compare the efficiency of these different types of circulatory systems. So metabolic rate itself is the quantity of energy used by the body over a given time. And that can be measured in kilojoules or kilocalories. So that particular measurement is taken from the fact that all of the reactions within a cell are using or providing energy. And we know that that's metabolism, the total of reactions within a cell. So those reactions are giving the energy from respiration, and therefore one of the methods of measuring metabolic rate is looking at how fast the products from respiration are produced and how fast the reactants from respiration are used up. So what we're looking at is glucose and oxygen being used up per unit time, or carbon dioxide, energy or water being produced per unit time. Now, glucose and water aren't all that easy to measure. How much glucose is being used up versus how much water is being produced are a bit more tricky to measure compared to oxygen or carbon dioxide. So therefore we tend to focus on the gases rather than glucose or water. We can also look at measuring the energy produced as heat within any given time to give us a measure of metabolic rate. Now, the methods for detecting or measuring these factors can range from very simple, low technology ones to more high tech ones that are going to give more real time and specific readings. A metabolic rate itself can be measured using either a respirometer or a calorimeter. A respirometer being one which will measure the rate of respiration, while a calorimeter will measure the number of kilocalories produced. So beginning with looking at a respirometer. And this is a common one that you'll see in various different exam, past papers, etc. It's quite a common experimental setup, would be one like this. And essentially what we're looking at is we've got a sealed chamber with a respiring organism present within it. We often also within this sealed chamber have something which is some chemical or substance which is absorbing our carbon dioxide produced by the organism. And the reason we're doing this is because what we're trying to establish is how much oxygen is being taken in by our organism. We also have a control setup which will usually contain some kind of object which takes up the same volume as the animal and the CO2 absorbent in the animal chamber, and that allows us to compare essentially it's controlling one of the specific variables. So the use of a respirometer is essentially really looking at the rate of respiration, so how fast that oxygen is being taken in by the. Alternatively, it is possible to use a calorimeter. And a calorimeter is going to measure the heat generated by an organism and calculate the metabolic rate from the results collected. So what we'd be looking at is how much heat can be generated by a specific organism as they carry out aerobic respiration. So in this diagram, you can see we've got our organism within a sealed chamber, and we have a pipe above with water in and water out. And what we're expecting to be able to measure is the heat generated by the organism. We'll raise the water temperature by a certain number of degrees, and we'd be able to measure that as the warmer water comes out, and therefore calculate the metabolic rate. You may also see this produces something like this. So we have our germinating pea seeds in one glass jar, boiled seeds in our control jar, so that they have been killed off, they're no longer carrying out respiration. And in our germinating seed jar, we're seeing a temperature raise with our thermometer. So we're seeing heat generated by these organisms compared to our dead organisms over here, obviously generating no heat. And again, the temperature raise can be measured in kilocalories or kilojoules depending on unit time. So what we're looking at is how quickly these seeds are able to carry out respiration in specific conditions. Now, to maintain this metabolic rate and maintain our respiration rate, essentially, we need to be able to provide oxygen to each of the cells that are going to be carrying out respiration. And in order to provide the oxygen to all of those cells, we need to have what we refer to as our circulatory systems within different organisms. If you have very simple organisms, they are able to, for example, use their cell membranes. So if we've got an amoeba or a single cell bacteria, they can use their cell membranes to allow for diffusion and movement of oxygen through their cell membranes. If we have a more complex organism, however, that's when we need to look at multicellular organisms using oxygen delivery systems. So the cardiovascular system that we can see in higher animals, and the sort of cardiovascular system that we're used to seeing when we studied at National 5. So it's important to realise that when we talk about the cardiovascular system, we're referring to some specific functions and some specific parts. So obviously we have the heart, usually made up of two different types of chambers, atria and ventricles. Atria being where the blood enters the heart, and ventricles where the blood leaves the heart. We also have blood vessels in the forms of arteries carrying blood away from the heart under high pressure. Capillaries being the smallest blood vessels where nutrients, gases and waste products between the blood and body tissue are exchanged. And we also have veins which carry blood back to the heart under low pressure. And that's really all you're expected to remember from National 5 at this point. Now we have different types of circulatory system which are referred to as either incomplete or closed, and they are referred to as being either single or double. So we have single systems where the blood is only passing through the heart once for each circuit of the body, double circuits where it's being passed through twice, and we also have incomplete systems where there's only perhaps one ventricle, for example, and we mix up some oxygen and oxygenated and deoxygenated blood. So within vertebrates, first of all our vertebrates have closed circulatory systems where blood is contained in a continuous circuit of blood vessels, and it's kept moving by the muscular pump of the heart. Now, within closed systems, as we said, systems are either going to be single or double as a reference to how many times the blood is actually pumped through the heart itself. Within a single circulatory system, for example, the type that you would see in a fish, it's referred to as our single system because the blood is passing through the heart only once for every circuit of the body. So the blood is flowing through the gills at high pressure, picking up oxygen and coming back through the capillaries, delivering that oxygen to all of the system, and the deoxygenated blood is returning back to the heart. And that's coming through at a relatively low pressure. And that's quite a primitive and very inefficient method of oxygen delivery. We may also have an incomplete double circulatory system, and this usually occurs in reptiles or amphibians. And they're described as incomplete because they only have one ventricle down here. We've got two atria, so it looks a very similar setup to the systems that we've seen previously. We've got our heart in the middle, we've got atria, we've got a ventricle. However, there's only a single ventricle, and that means that some oxygenated and deoxygenated blood is mixing within this ventricle. It's got a very similar system though. We have got our blood, our oxygenated blood, leaving the heart, coming round into our system, into the capillaries, and releasing the oxygen into the cells around the body systems and cells. That deoxygenated blood then flows back into the heart, where it's pumped back out to get to the lungs or perhaps to the skin capillaries, and we'll come on to those in a second, where we're able to collect oxygen before it's returned to the heart to be pumped around again. So within our amphibians, that mixing of oxygenated and deoxygenated blood in the single ventricle isn't actually really a major problem, as the blood returning from the body has already been partially oxygenated through its moist skin. So if we go back to have a little look, we've got, as I said, we've got skin capillaries as well as lungs which are able to collect oxygen, but also as the blood has been pumped out, oxygenated blood has been pumped out and into the system, as it's flowing back, or in fact as it's flowing through here, we have the capability, although this is deoxygenated blood returning from the systems, we're able to pick up some oxygen from the skin at this point as well. So some of this will actually be oxygenated blood coming through. It's predominantly deoxygenated, however, because they have really moist skin, and skin which is permeable to oxygen through here, it's possible that some of the oxygen is able to get through and be picked up by those veins as it returns to the heart. Finally, in birds and mammals, we have the complete circulatory system. So that's referred to as being complete because we've got two ventricles completely separated by what we call the septum. And it's the most efficient and most advanced circulatory system, and that allows our endothermic vertebrates, so vertebrates which are able to generate their own heat, deliver lots of oxygenated blood to the respiring tissues, and also able to pick up the heat released by those tissues and circulate it. So again, just as what you've seen previously at National 5, we have our oxygenated blood being pumped out of the heart around to our body systems. Within the capillaries there, we're passing off the oxygen into our systemic cells, and that's allowing respiration to occur. We're then able to pick up the carbon dioxide and waste products, take that back to the heart where it's pumped back out to the lungs. We're collecting oxygen, and for the cycle to start again. So this is referred to as our complete double circulatory system. Complete because we don't have any mixing of oxygenated versus deoxygenated blood, and a double system because it's pumped around twice. And all of these types of circulatory system are the methods by which we're able to maintain that metabolic rate within different organisms, providing oxygen to the respiring cells to maintain that metabolic rate. So from that, you should be able to list the possible measurements used to compare metabolic rates. The fact that we can measure the amount of oxygen used up, the amount of heat produced, or the amount of carbon dioxide produced, for example. That different organisms have different metabolic rates. So you should be able to see that we have various measurements in order to compare different metabolic rates. We could be looking at the oxygen used up, we could be looking at heat generated or carbon dioxide produced. We can also look at how different organisms have different metabolic rates. The fact that those which have faster rates of production are going to, or faster or more energy requirements, are going to have a higher metabolic rate. that we have different circulatory systems used by different organisms. So we have a single system, a double system, and we have closed and incomplete systems. So the circulatory system as a fish is our single system, and blood passing through the two-chambered heart only once for each circuit of the body. And within reptiles and amphibians, we have a double incomplete system because it's going through the heart twice, but it's incomplete. There's some mixing of oxygen and deoxygenated blood. And lastly, we have our complete double circulatory system seen in birds and in mammals, where the blood is passing through the heart twice and is completely separated. We have no mixing of oxygenated and deoxygenated blood, and that this is the most efficient type of circulatory system.